Thank you very much, John. Thank you for the invitation, and thanks for coming to listen to me blab a little bit about some of the stuff that we're interested in. And so, as John mentioned, I'm doing my work here at Western. Okay, so there's a pretty picture of Western. It looks nothing like that this morning when I left. Oh yeah, so there's the running joke that that my place of employment is the University of Western Ontario, but they decided a couple years ago to change the name. I'm not really sure why they did that, but because I'm not really sure exactly what we're west of or east of, but we're now known as Western, so that's where I'm from. So what I want to talk to you a little bit about is to sort of set the stage in terms of why I'm interested in doing the work that we do. And really at the end of the day, I'm interested in understanding what makes the brain vulnerable, what makes it vulnerable to different types of diseases, what makes it vulnerable to different types of injury, why do different regions of the brain respond differently than other regions. So for example, in Parkinson's disease, why is it the substantia nigra that's the most sensitive to cell death? And in Alzheimer's, why is it the basal forebrain? And ischemia, why is it the CA1 of the hippocampus? So what makes the different brain regions more vulnerable? And the common factor to all these things is really age. And we now, as this is not news to anybody in this room, but really we're dealing with an aging population. If you look at the level of the demographics over the next 20 years, we're really in a situation where we're dealing with a lot of fragile brains, and I think that's kind of the precipice of the research program that I'm interested in. And at the end of the day, you know, one of the most tragic things that happens to people when they get older is this risk of developing dementia. And one of the things that I'm going to be talking to you about a little bit today is that dementia on its, Alzheimer's disease and dementia doesn't really happen on its own. Usually there are other comorbidities that are present. In fact, stroke is one of the strongest comorbidity, one of the biggest cofactors in influencing Alzheimer's progression. And roughly 1 in 3 to 1 in 4 people have a lifetime risk of developing dementia, but 1 in 3 or 1 in 4 will have the risk of developing either stroke, dementia, or both. And this idea of what happens following a stroke in terms of what happens to the brains is actually particularly interesting. So turns out about 1 in 4 to 1 in 5 patients that come into a stroke clinic will go on to develop some type of dementia, some type of cognitive impairment and dementia. Some will be Alzheimer's type, some will be vascular type, some will be mixed. And really, it's hard to sort of distinguish what's what. But at the end of the day, some patients are going to go on to deteriorate. So we're left, I've always been intrigued with the question is, why do some patients go on to develop post-stroke cognitive impairment while others do not? And so that's sort of one of the things that sort of got me really interested in this. And I know you're saying, well, I thought I came to talk about lipids. And I am going to talk about lipids, but the broader picture of my research program really is interested in looking at this idea of comorbidities and understanding why different brain regions are more or less vulnerable to attack, and that's kind of where the lipid story fits in. So just bear with me a little bit as I take you on this journey. So you may be asking yourself, well, I didn't even realize there was any kind of evidence that AD and stroke actually interacted, and actually it's true that it does. Back in the 80s, 70s, 80s, 90s, early 2000s, there was a study where they followed a group of nuns, and why they chose these nuns is because they have similar environmental controls. They all get up at the same time, they all eat the same thing, they all pray at the same time, they're all exposed to the same environment. And what they found was that at autopsy, they followed the behavioral cognitive impairment, and they matched what happened at autopsy. And they found something really interesting. They found that if the nuns had evidence of a cerebral infarct, as well as some level of Alzheimer's disease pathology, that the level of dementia was roughly 30 to 40% worse in these patients than if they only had only the Alzheimer type dementia pathology alone. What was even more interesting is if that stroke was a subcortical lacunar infarct, and to set the stage, if you're not aware sort of the kinds of strokes, subcortical strokes are kind of like those transient ischemic attacks or those silent strokes that happen and patients don't even realize that they have, but then they start to develop cognitive impairment 5-10 years down the road. If they have evidence of these subcortical strokes with some level of Alzheimer pathology, the level of dementia goes up 80 to 90%. So there's clearly some interaction, and it's not just an additive effect between the impairment that's happening due to the stroke and then the impairment that's happening with the Alzheimer pathology. And so other studies have also followed up, and we've done some work in the lab since then. And if you look at the area of tissue damage caused by a stroke, or which is called a stroke-induced infarct, and you look at some of the biochemical changes, you can actually see some of the pathological precursors, some of the histological precursors associated with Alzheimer's disease in this tissue area. And it kind of also makes sense that these two might be interacting, because the same risk factors for having a stroke are more or less the same risk factors for developing Alzheimer's disease. Hypertension, type 2 diabetes, atherosclerosis, smoking, obesity, age. All of these are common risk factors for developing Alzheimer's disease as they are for developing, or for the chances of having a stroke. And also, there's been research that's shown that if you have Alzheimer's disease, the risk of stroke basically triples. And if you've had a stroke, the risk of developing Alzheimer's disease essentially triples. So there is some clear interaction going on between the two. And the question is, what is the interaction? And so in Alzheimer's disease, there's two main pathological features of Alzheimer's disease that happen. One is that you get the abnormal accumulation of this beta amyloid protein into plaques. There's a lot of controversy into the field as to what's the toxic substance, but from a standpoint in terms of a clinical diagnosis, you need to have the features of these amyloid plaques that are deposited, as well as hyperphosphorylation of this tau protein forming this neurofibrillary tangles, and then of course you get concomitant cell death. But what also happens in these Alzheimer's disease patients is they get a strong neuroinflammatory response. So what happens is the A beta gets deposited, and then activated microglia recruit to the site, astrocytes. They try to remove the plaque material, they try to remove the beta amyloid, and they just can't do it. They are really poor at clearing this material. And so what happens is the amyloid persists, the inflammation persists, and instead of having an acute inflammatory response, you end up with a chronic inflammatory response. And it seems to drive the whole process forward. So the neuroinflammatory response is a key component to the Alzheimer's disease pathology. And what's interesting as well is if you look at the stroke and what happens in terms of the pathophysiology of a stroke, normally you get a restriction of blood flow to a specific area, either caused by a clot or a hemorrhagic stroke. The majority of strokes are this ischemic or clot-induced strokes. But one of the things that's interesting about this about the stroke is that a lot of the delayed neuronal deaths, so the tissue at risk outside this stroke-induced infarct area for future cell death is really under the influence of oxidative stress and neuroinflammation. So neuroinflammation does seem to play a key role in the pathogenesis of controlling the potential outcome, or how worse a patient's going to do following a stroke. So some of the work that really got me interested in this as a student, as a postdoc, was, well, you know, if inflammation is involved in Alzheimer's and inflammation is involved in stroke, then maybe this is one of those mechanisms that causes the interaction between the two of them. And this kind of makes sense. So if you have a brain that's in a vulnerable state, somebody's had a stroke, you get some inflammation there. And the inflammation, we also know, is really important in driving Alzheimer's disease pathology. Is it possible that that inflammation you got from the stroke or hypertension or diabetes is priming the brain and driving the Alzheimer's disease progression even further? Or is the other opposite true? Maybe you have beta amyloid deposition in the brain with very low, but you don't have cognitive impairment, or you just have that seeding level where the Alzheimer's disease pathology is just starting to happen, you're getting some inflammation, the brain's primed again, and then you get a stroke where you cause a massive inflammatory response. Maybe the interaction of that enhanced inflammatory response is enough to tip the patient over the edge into developing full-blown Alzheimer's disease. So this is the work that I started doing my PhD and postdoc, and I'm not going to spend too much time about it because it has nothing to do with lipids, but what we modeled in the lab to be able to do this is for our animals where we gave a stroke, what we did is we injected this compound called endothelin, which is a potent vasoconstrictor, and we put it into one side of the brain, into the striatum. And the reason why we chose the striatum is because, again, this would mirror those subcortical locations that you were seeing in the Nun study. Remember, it was the subcortical strokes plus the Alzheimer's pathology that would drive the overall response. So we would do that, and then what we would do is we would infuse or inject beta amyloid into the ventricles bilaterally to induce the Alzheimer's disease model. Now, I use the term model loosely. This is a non-transgenic model, obviously. This was done a long time ago. We're now doing a lot of this work in transgenic animals, but this is one of the first proof of principle studies to look at the actual interaction. What's nice about injecting the amyloid into the animal is that you can control the onset, right? So the problem with the transgenic animals is they start producing these unphysiologically relevant levels of beta amyloid because you're basically putting in a mutated human gene into a mouse or into a rat, which we're now doing, by the way. But you know, it's really hard to look at early stages, early inductions, because you're talking about a genetic model. And that only really accounts for 5 to 10% of all Alzheimer's patients, where the other 90 are sporadic or unknown onsets and causes. So this is how we induce that model. And then what we would do is we would induce both at the same time. So we would have some animals where we inject the beta amyloid, and then we have some, and we'd also give them the stroke. And then when we look at the interaction, what we found out was really interesting. So we did a number of studies looking at this. And so what we're looking at here is we just asked the question is, if you put amyloid into the system, so the Alzheimer's pathology into the system, and then you induce a stroke. Okay, so this is asking the question, does the stroke push the animal over the edge? Or is there some type of interaction between the two of them? So what we decided to do was just look at the area of damage caused by the stroke and ask the question, was there some kind of interaction between the Alzheimer pathology that we induced and the stroke that we are inducing? So we measured the infarct using just a simple Nissl stain. And what we're doing, what I'm showing here is in the black bars is these are the stroke alone animals, and we measured the infarct volume at 7 days, we measured the infarct volume at 28 days, and over time we actually found that the infarct shrunk. And this has also been shown clinically, so this was interesting. However, when you added the combined paradigm into the system, not only did the infarct not shrink, but the infarct grew over time. So in the presence of amyloid, the infarct doesn't shrink, it actually grows over time. So there's some type of interaction. Now remember, we put the amyloid into the ventricles, right? So we're talking about it's not because of the juxtaposition of an anatomical phenomenon. There's something else going on. You know, maybe it's the inflammatory response. So we did the exact same measurements looking at activated microglia here and reactive astrocytes, and when we quantified it, we found the exact same pattern. We found that over time, in the presence of amyloid, the stroke-induced inflammation also increased over time, as opposed to resolving itself. So clearly there was, from these basic animal studies, there was some indication that there was an interaction between the stroke and the Alzheimer's. So then okay, well that's great, that's what's happening in the stroke. So since we've also looked at some brain regions remote from the actual infarct. And so here we're actually measuring A-beta deposition in our animals. Again, we have the uninjured animals here on the bottom. We have the stroke alone animals here. We have the A-beta alone animals, and then we have the A-beta stroke animals up here. And here we're measuring amyloid deposition. It has nothing to, has nothing to do with anatomical position. It's widespread all throughout the brains of these animals. So then we asked the question, well, was there some kind of functional problem due to this interaction? And so what we did is a test called the circular platform test, and it's kind of like a land-based Barnes maze. And what you do is you put a rat on a table and you look at, and in one of the holes you have a drop box, and the rats will instinctively want to go in the drop box, and you train them over time and they can do the test and they become very good at actually finding the hole location. And then what you do is you give them one of the surgical paradigms, either the stroke alone, the Alzheimer's paradigm alone, or the combination, and then you basically test them a period of time up to 28 days following inducing those surgical paradigms, and you're asking the question, do they remember where the hole location is? Very, very simple test. Then what you do is you really screw things up and you move the hole location on these poor guys, and you ask the question, can you relearn the same task on a new hole location? So we're testing reference memory, we're testing spatial reference memory, and then we're also testing learning and the ability to learn the same task but on a different location. All right, so this is what the data looks like on an animal that hasn't been injured. So you can train them on the y-axis here. We're looking at time to find the escape hole. And here's your pre-surgical trials. And you can see that over time, the animals become very good at finding the hole location. And this point right here on trial 15, this is us asking the question, do you remember the original hole location up to 28 days after a sham surgery? So basically in these animals, we put them under the anesthetic, we open up the skull, we drill holes, but we don't put anything in the animal. And then we do the reacquisition where we move the hole location and we ask them, can you relearn the task? And yes, they can relearn the task. And in fact, they're better at doing it the second time than they are the first time, which kind of makes sense, okay? So what happens if we give them the A-beta or the Alzheimer alone? Well, there's your learning curve. Obviously, nothing's happened to the animals yet. There's us asking the question, do you remember where the hole location is? Generally speaking, there is some impairment. Obviously, the longer the amyloid's cooking in the brain, there's a greater impairment. And then we ask them, can you relearn it? Yes, there is some impairment in the older animals, but generally speaking, they can relearn the task, okay? There definitely is some impairment for sure. if we add both the stroke to the Alzheimer paradigm, they all have a hard time remembering where the whole location. They all have a hard time relearning it. So not only is the stroke-induced infarct growing over time, not only are we seeing remote changes in inflammation and remote changes in amyloid deposition in these animals, but they also have a behavioral consequence to the interaction between these two conditions in the animal model. And so what this led us to is to potentially come up with a schematic and something that we continue to work with now and look at, well, where are the potential points of interaction? And so amyloid is normally generated through this precursor protein, and that itself can drive more amyloid deposition. We showed, although I haven't shown you this data, but we've published on it a number of papers on it where we showed that this beta amyloid can cause a strong inflammatory response leading to upregulation of inflammatory cytokines and transcription factors, and this in itself can drive forward this loop and again sort of increase the feed-forward mechanism of the disease. And then when you add in the stroke, we also showed that stroke can increase the pathological correlates of Alzheimer's disease and can also drive the inflammatory response. So again, the question became over the timing, when do all these things happen? So in this particular animal paradigm, we induce everything at the same time. What would be really interesting to know is if you pre-treated the animal with the amyloid for some time and then gave the stroke, or induced the stroke and then induced the amyloid, and started playing around with the timelines. So that's some of the work that we're currently doing right now, as well as investigating this particular interaction in transgenic animals. And so the reason why I wanted to spend a few minutes showing this is because this type of interaction really sort of set me on the path of my research program and some of the stuff that we're doing now. And so the first theme, which I'm actually not going to be spending any more time talking about, but I just want to kind of set the stage, is looking at the interactions in the vulnerable brain and continue to explore the relationship between stroke and Alzheimer's disease, but also explore the relationship between hypertension and Alzheimer's, type 2 diabetes and Alzheimer's, because really stroke is just the end point of vascular disease in many cases, or cerebral vascular disease. It's what's happening 20 or 30 years later that leads up to that point. So what's going on in hypertensive individuals? What's going on in type 2 diabetic individuals? And trying to, again, assess the role of inflammation, but as I'm going to be talking about later, is also assess the role that maybe membrane biochemistry might be playing in all these things. So asking the question, why do local injuries cause global responses, and then developing animal models to be able to appropriately answer this question. So one of the things that we've actually just started acquiring and have been aging a colony of transgenic rats that have the human APP mutated gene in it. They produce beta amyloid, but they're really good at clearing it. Okay, these animals they don't develop plaques. They have, after 1 year, they show mild anxiety and mild executive dysfunction, which is kind of interesting because those are the earliest phases of symptoms that you will see in an Alzheimer's disease patient. But they don't develop the pathology unless you trigger some type of stress in these animals. So with the stresses that have been shown, if you give this animal traumatic brain injury, guess what? It tips it over the edge and they start developing Alzheimer's pathology. The work that we're doing is we're inducing stroke in these animals. We're also inducing hypertension and type 2 diabetes in these animals, and we're going to basically see the mechanistic interactions. We just started this study a year ago. It's an extremely expensive study to do because we have to breed all the animals. It's not like breeding mice. Breeding rats is way more expensive. And then we also age all our animals up. We don't even start to do anything into these animals until they're at least 6 months of age. That's actually one of my, on a side note, it's one of my biggest pet peeves about animal research when it comes to neurodegenerative disorders. Everybody does things in 8-week-old animals. And that's like the equivalent of doing something in a 10-year-old child, right? You just can't equate. If age is your biggest risk factor for neurodegeneration, why are you doing something in an 8-week animal? Anyway, I get why we do it because it's cheaper and it costs a lot of money to age these animals, but you know, are you really getting the right answers to the questions that you're looking for? So this is something that we're very aware of and we're sensitive to. So some of the things that we're answering these questions is we started some translational research where we're confirming some of these experimental findings in humans, actually. So we want to confirm in humans that these cerebral infarcts, so that these infarcts following a stroke grow in the presence of amyloid. And we also want to confirm in humans the experimental findings that the infarcts in the presence of amyloid produce greater amounts of inflammation. So basically what we want to do is it's kind of we say, hey, we found this really cool thing in our animal models. Is this really happening in humans? We think it is based on some of the preliminary evidence that people have published, but it's never really been directly looked at. So we're doing that. And then the idea is that with the help of the animal models, we will go back once we've confirmed it in humans, which we're doing right now, and then start to go back into the animal models and start teasing apart the temporal profile of these things, of how they're happening. And so getting back to the original question of why do some patients who show up who have a stroke go on to develop dementia and some do not, these types of studies are going to help to answer those types of questions. Maybe there was some underlying amyloid, because we all produce amyloid in our brains. Some of us are better at clearing it than others. Maybe the stroke is just enough, or the hypertension or whatever is just enough to impair the clearance and to add the inflammation to push the patient over the edge. So then the question becomes, can we use A-beta therapy and anti-inflammatory therapy, not in Alzheimer's disease patients, because by the time that's diagnosed, it's too late to do any of this work. But can we do this in a cohort of patients who've just had a stroke and prevent the secondary decline 10, 15 years down the road? So that's the thing that we're saying. So this work is funded by my CIHR grant, and it's currently underway. And we do have some preliminary evidence in the human population. So the way we do this is we do PET or MRI imaging of beta amyloid, and we can do the A-beta amyloid imaging using this compound called the Pittsburgh B compound. And then what we can do is we can also use a compound called PK1195, and this is with a collaboration of a team of investigators at McGill. And what we do is we can use this PET ligand to label activated microglia. So we're asking the exact same questions, but we're doing it in human patients. And here's some preliminary data that came out a few years ago, and we're following up on in our animal models. And so here's a stroke patient here, and you can see the inflammation here caused by the infarct right here. And what was really cool about this actually is that the infarct is unilateral, but the inflammation is bilateral in the white matter. I don't know if you can see it from back there. And then if we image that exact same patient at 6 months and you look for the infarct, it's gone. The inflammation is completely resolved, as our animal model showed, right? But the inflammation persisted in the white matter fiber tracts 6 months later. Unfortunately, we weren't able to do the CPIB because we didn't have the approval to do it then, but this is the kind of studies that we're doing in the human patients right now. So we're doing the PET MRI imaging in the human patients following stroke, and we're also doing the same type of imaging in our rat models following the stroke with and without the Alzheimer's disease paradigm. Again, the idea is we can use the animal model to come up with a therapeutic window of opportunity to find out the temporal and spatial profile of inflammation and amyloid deposition, and then hopefully go back into the humans and use that therapy in the human patients. So what's really nice is that the investigators that I'm working with on this, we have a nice team of clinicians and we were talking about this at lunch. It's a nice a team of clinicians and basic scientists working this together. And everybody talks about doing translational research, but it's really actually hard to do, right? Because there's egos involved, and you're working with clinicians, and they're not always the easiest people to work with. And basic scientists can be a real pain in the butt too. And but it's nice because we, so my postdocs and their fellows, we have weekly meetings, right? And we talk about this kind of stuff. And their fellows come and do wet bench research in my lab, and my postdocs get some exposure to some clinical work too. So it's a real nice environment that we've set up. Okay, so at the end of the day, what we're asking again is looking at this reciprocal interaction between stroke, inflammation, and amyloid, and asking the question that if suffering a stroke represents a tipping point in a cognitive decline with patients with an amyloid load, then maybe treatment at the time of a stroke could make a difference. And again, this is the work that we're looking at. So that's where we're at with that theme one. And unfortunately, I don't have a lot of preliminary data more than I showed you right now, because we just got that grant work funded and we're just starting the work now. So hopefully in a year or two, we'll have some positive results, or at least some results to show you on that. So I want to spend the next time is shifting focuses a little bit, but same type of idea, and drilling down to a more biochemical way of answering the questions. So yes, inflammation does seem to be mediating the two pathologies, but is there something else maybe more intrinsic that could explain the vulnerability, the differential vulnerability of the brain? So again, going back to the question, what makes different brain regions more or less vulnerable to attack. All right, so if you consider, that many neurodegenerative diseases have common molecular pathways, okay, but have distinct anatomical connectivity, right? Are there common molecular pathways that could potentially, or changes within the anatomy that could partially explain why different brain regions respond differently following attack? So again, to explore what makes different neuronal populations more vulnerable to different types of attack, whether it be stroke, A-beta, Parkinson's, whatever. And then that kind of led me to this idea that, well, maybe all things being considered, what's the first line of communication with a neuron in its external environment? That'd be the plasma membrane. You think, well, yeah, there's different receptors. They have different things that are expressed, and that's absolutely true. But things organize themselves very differently in membrane domains in different regions of the brain. So it turns out that if you look at the brain as a heterogeneous structure, it's put together very different from a lipid biochemistry point of view, depending on its anatomy. And if the membrane is so important in things like signaling, fluidity, proliferation, apoptosis, all these things that you can think about, maybe there's some intrinsic property to the plasma membrane itself that could partially explain or influence the level of vulnerability to different types of attack and injury. So that's what we want to do. So the first thing that led me to this story was, well, and this was when I was at the NRC and have continued on here at Western, is that I was very intrigued by these lipid rafts, right? And there wasn't a lot known on them, and there's still really not a lot known on them. They're kind of one of these things that is extremely controversial because really they're hard to see. You can't really label them that well, and you don't really know what goes on inside them. But the membrane is divided into different domains, and you can have cholesterol-rich microdomains that influence level of endocytosis, signaling cell structure, signaling all these different types of things. But what was really interesting, the more and more I dug in the literature, is it found that if you look at the membrane raft composition in different areas of the brain, it was heterogeneously expressed within the brain. So different brain regions had different levels of lipid raft activity, which made me think, well, maybe actually the lipid biochemistry is different in these different regions as well. So that's the first thing I started chasing. Well, maybe they have a role in vulnerability. Maybe these lipid rafts are really important. So here's some studies that we did. And actually, this is some unpublished work that I've never was able to follow up on. And what we're labeling here, I don't know how well you can see it at the back, but we're labeling here with the substance called collotoxin B, which labels a ganglioside called GM1, which is enriched in lipid rafts. And then here we're looking just at F-actin. And these are like early cortical neurons, so like day 2, day 3 in vitro. And then we looked at the lipid raft activity, and what we found was that they were really highly expressed or highly labeled, obviously on the cell surface, but at cell-cell junctions. So that was interesting. But they were also, when we looked back early, they also seemed to be localized at areas where the axon was going to extend. So I've never followed up on this, and I show this, and people are interested in it, but I've never seen anybody really follow up on this on the stuff. We've never published on it, but we think that, based on some of this work, that the lipid raft organization, that these lipid microdomains might be playing a really important role in determining polarity. And it turns out that we've done some experiments where you can disrupt the lipid raft activity and then allow it to reorganize and then change the polarity of the cell based on just allowing the membranes to reassociate. So there might be something to this. That's not really where my research interests lie. It was just some kind of anecdotal evidence that we found. But it led us to the question, okay, well, maybe based on this stuff and based on some other work that we published that demonstrated that lipid rafts could organize different proteins involved in injury and that kind of stuff. Like following a stroke, for example, a lot of proteins, glutamate receptors, all these things get shuttled to lipid rafts and enhance the excitotoxic response and neurodegenerative response. So then I asked the question, well, if you could somehow prevent all this from happening, maybe this would be an effective neuroprotective strategy. If you could prevent these lipid rafts from doing their job following injury, maybe this would be an intrinsic way to protect the cell from dying. Well, it turns out that I was completely wrong. So what this experiment showing is here is we have primary cortical neurons, and this is live cell imaging, and we are treating, and we've done this with different concentrations of this agent called methyl beta cyclodextrin, which is basically a cholesterol depleting agent. And what the theory is that it would remove the ability of neurons to form these microdomains because they don't have the cholesterol to be able to form them. And then the idea is, well, let's just throw this stuff on and then like give them oxygen glucose deprivation, a stroke in a dish or something to kill them and see if it protects. Well, it turns out that if you just give it to them on its own, this is what happens. They don't like it. And in fact, they start to retract. Okay? Notice that the cells are still alive. Okay? But did you notice the neuritic network is completely gone? And it's almost like they're becoming, and look at this guy, it's like it's very organized. It's being shuttled backwards into the cell. And if you look at, again, if you look at, let's see if I can pull that up. If you look at the architecture of all the neurites there, and then you give them the agent, again, they retract very quickly. And they get sucked back in the cell, and they form like this sphere-like cell body, right? And if you ever plated primary cortical neurons or hippocampal neurons, like when you first plate them in, they look like stem cells, right? And that's kind of what they look like before they extend their processes. It's like almost like it's gone in reverse, and everything just, in the absence of lipid rafts, everything starts to collapse. And these cells, and what happens is that if you repeat the experiment and you give propydium iodide, so what propydium iodide does is it'll get taken up by cells whose membrane starts to become leaky, who are undergoing neurodegeneration, and you do the exact same experiment. Again, what I wanted to highlight is that the neurites all start to retract prior to the cells undergoing cell death. So they're still alive. The neurites are retracting. They're not dead yet. Okay, yeah, some of them exploded, but for the most part, they're still there. And it's not until the neurites all start to retract that they start to glow red with the propydium iodide, which you'll start to see very soon. Just wait for it. Still going. They're still alive. I mean, they're not happy, but they're not dead, right? And then they start to express the propydium iodide. So the neurites retract, and we actually quantified this looking at time-lapse imaging. And what we did is we looked at neuritic retraction. So here we're looking at, in red, we're measuring the overall average length of the neurites. And then in here, we're using two different markers for looking at neurodegeneration. And we showed that the neuritic retraction always preceded the cell death. So why was this interesting? Well, first of all, obviously, lipid rafts are important, that you can't completely inhibit them. But and if you do, the function of these guys is really important for maintaining potentially cell structure. So some of the data I don't have time to show you is that we also followed up looking at, oh, actually, I think I did include the slide. Yeah, I did actually. Sorry about that. So what we did is we co-labeled these repeated the experiment. We co-labeled them with different microtubule-associated proteins, so different cytoskeletal proteins. We looked at like tau, microtubule-associated protein, beta-3 tubulin, all the different cytoskeletal proteins of the neuron. And then we co-labeled with this, the lipid raft marker, and we did the same thing. So this is what it looks like. You kind of get neuritic expression of lipid rafts and cell body expression of lipid rafts. But what happens is there's roughly equal amounts of lipid rafts in the neurites as there are in the cell body. And then what happens is if, and this is smalls, but I will highlight some of the pictures below. What happens is if you repeat that experiment with the cyclodextrin and look at the labeling, the lipid rafts become very, very disorganized. And then what happens is they start to localize very strongly into the cell body and outside of the neurites. So what we think might be happening, and based on this evidence, is that if you disrupt the lipid rafts altogether, what happens is they lose their localization in the neurites. They get shuttled to the cell body. They get clogged up in the cell body, and as a consequence, the neurites start to retract. Okay, and so what happens is that we think is that not only are these lipid rafts really important for signaling events in the cell body, but they might be actually acting as anchors and neurites for microtubule-associated proteins and acting as cytoskeletal support network, as a physical support network. And when you lose that physical support network, then things start to retract. Okay, so then you're asking, well, who cares? Why should I really care about this? Well, if you go back to my first point and you're looking at vascular health and neuronal function, right? things like fatty acids, cholesterol, all these things which can influence lipid raft activity, the ability to assemble, the ability to form potentially this architecture. If you can somehow shift that even subtly by 10% one way or another, right? Can you change the synaptic communication between one neuron and an adjacent neuron because you've changed the ability to form these networks? Right? So we showed the drastic case. I mean, you completely remove cholesterol from the system, this is what happens. But if you can, say, adjust cholesterol 5% or even 1%, what would be the outcome on the cell architecture? I think that is the real interesting question, and why that's applicable to some of the other work that we're doing is things like diet and exercise. All these things can affect central lipid levels, right? Which potentially have a direct consequence on cell-to-cell communication. All right. So the next question I want to finish the last part of my talk with, and I think this is the real exciting thing, is to talk about how we can visualize lipids in situ, in tissue. So why is this important? Well, based on the evidence that I showed you before, lipids and what's contained in the different microdomains is probably a really, really important phenomenon. We don't know a lot about it. Why? Because you can't actually physically visualize. There are very few techniques that you can use to look at lipids. So unlike immunohistochemistry, where you're tagging peptides, you're going after a specific epitope. Well, what would an epitope be in a lipid? Right? Chances are that same epitope would cross-react with thousands of other lipids, and that's the problem. You can't do immunohistochemistry based on lipid biochemistry. So you have you have different, basically you're going after a head group. And the other thing about lipids is that they differ. The same lipid can differ in its fatty acid chain length, or it's like you can have the same lipid with 18 carbons, the same lipid with 20 carbons, right? And maybe they have different functions, maybe they don't. There's no way to actually detect that with normal visualization methods. And I always say, my adage is that, it's great to get biochemical readouts of lipids and stuff like that, but really a picture can tell 1,000 words, right? So you look at here, and you don't have to be a lipid biochemist to know that something happened, right? And these are 2 of my 4 kids, and I'm not, we're not really sure. One looks really guilty and one looks really sad, right? So you can just infer by a picture of what's going on. I think that's what's missing in the lipid biochemistry world is this positional information, this anatomical relevant positional information. And so what I started to do when I was at the NRC and continued now is a big focus of my research at Western, is to develop technology with people who are mass specs. So I was asked the question at lunch, are you a mass spec guy? I'm not a mass spec guy by any means. I'm interested in using mass spec to answer questions. So I partner with people who are mass spec people. They're interested in developing methodology. I'm interested in answering questions. We work well together. Okay, so I decided to say, well, let's go after some lipids that we think are important to some of the research questions that I'm interested in. I said, well, a good class of membrane lipids to go after are gangliosides. Why? Because GM1 is very abundant in the brain. Turns out that it's a major component, as I showed you with that collar toxin B, of lipid rafts, right? So you're kind of just sort of weaving you through the story of my life and why I decided to look at these things. And it turns out that there's actually a really important story now that we're getting some data on it. And it turns out that these gangliosides and derivatives rapidly turn over in response to change and to injury, to cellular response. They're potentially heterogeneously expressed in the brain. So maybe they're one of these good targets to at least start with, right? So instead of looking at a needle in a haystack, let's just pick the needle and forget about the haystack and focus on what these guys do in the injured brain. And that's exactly what we did. So we looked at a middle cerebral artery occlusion model of a stroke in a mouse, and we looked at different time points following the injury, and we followed up the changes in ganglioside imaging. So again, just to give you some very, very basic biochemistry, this is how these ganglioside molecules are put together. You start with a sphingosine base, and the sphingosine base can have variable carbon chains, so you can have 16, 18, 20, 22 carbon chains on it. And then you add a fatty acid chain to it to get a ceramide molecule, and of course ceramides, as we all know, is a very important signaling molecule. And then you add a whole bunch of sugar residues, and you add them kind of on this train track, and the more sugar residues you have, the bigger the ganglioside molecule. So it's a nice attractive target to look at because it has different mass sizes. It has, it starts with a precursor and can start to build up in terms of building blocks and through different enzymatic reactions. So how do we do this? Well, we use a mass spectrometer called the MALDI-TOF, which is matrix-assisted laser desorption ionization, time of flight. And it's a technique that is used to analyze molecules. And the way what happens is you have a sample and you ionize it with some type of ionic matrix. You take a laser, you shoot the laser on it, it ionizes the tissue, the ions fly out, and then you calculate the time it takes to fly to your detector tube, and that's basically MALDI-TOF 101. Okay, so the bigger the molecule, everything has a different unique time of flight property. The bigger the molecules take longer to fly, and then you can put together the mass based on the time of flight algorithm. Again, it's more complicated than I understand, but apparently that's how it works. So how do we do this in tissue? Well, you take your animal, this is, you know, this could be any of the animal models that we showed before, or in this case, we just gave this a middle cerebral artery occlusion, which is a stroke model. And then what we do is we sacrifice the animal, we section the tissue, fresh frozen. And by the way, this isn't trivial to section 10 micron sections on non-fixed tissue. It's very tricky to do, and my grad students have become very good at it, mostly because I look over their shoulder and yell at them if they get it wrong. No, I don't yell at them. But then you put them on a MALDI plate, and you spray it with an ionizing matrix, and then you blast it with a laser, and then the ions fly off the tissue. And what you do is you blast it with a laser, kind of like a dot matrix printer. So you hit it in an ordered array in an XY coordinates, and then every coordinate is going to give you all the molecules that are flying out. So it's going to give you all the mass spectrum of anything that you're interested in looking at every positional information, or every position. And then you use some fancy computer software that, again, I don't know how it works, and you generate mass spectrum for every point, and you can put it all together and get an image. Actually, I do kind of understand how it works, but really, I don't understand the algorithms. And so here's an example of what a MALDI image will look like. Then what you can do is you can highlight regions of interest in this. So on this animal, this is the stroke side, this is the non-stroke side, and you can calculate the regions of interest, and you can use algorithms to calculate the area under the curve to do semi-quantitative analysis between the amount of species that's here versus the amount of species that's there. So in real life, this is what that data looks like. So here's an adjacent section, a crescent violet section. Here's what the MALDI image, the mass spectrum looks like. And if I just select regions of interest from the hippocampus, the cortex, and then here into the basal ganglia and the striatum, you can see that you get mass spectrum for each region of interest. And so then if I just want to look at what does the image, where is all this GM1 located, I can then pull up an image from that same tissue section, and anything here that's white is where GM1 is. And then I ask the question, well, what if I look for the same molecule, but the only difference is that this GM1 has 18 carbons on its sphingosine chain, and this one has 20 carbons on a sphingosine chain. This is the same tissue section, okay? The only difference between this molecule and this molecule is that this one has 20 carbons on its sphingosine chain, this one has 18. And look at the anatomical distribution, completely different. And if any of you have any interest in hippocampal and neurogenesis and that kind of stuff, you'll notice that here, this region here is a dentate gyrus. When this is one of two regions in the brain where you get new neurons that are born, that you can actually have neurogenesis that's occurring. And that's the region where you have pretty much exclusively the 20 carbon. So again, you're like, why the same ganglioside, 2 carbon difference? Why is it, why do they have different anatomical distributions? I don't know. But it may at least partially help us explain, well, guess where the one region where you don't get amyloid deposition in the Alzheimer's disease brain? In the dentate gyrus. You get it everywhere else in the hippocampus, but not there, right? So it's interesting. I mean, obviously that's a huge leap of logic, right? But not really. So we can start to answer those types of questions. So this is what it looks like. We did the experiment, and we followed different time points following the stroke. So here's an uninjured animal. Here's a stroke at 24, 3, 7, 14, and 28 days. Obviously, these guys are different animals because this is all done post-sacrificing tissue. But this tissue section is the same as this one. And we're looking at just GM1, the 18 versus the 20. Remember, this is the ganglioside that's upregulated, that's heavily associated in lipid rafts. We don't know if the 18 or the 20, if one is more in lipid rafts than the other. We'd love to be able to do those experiments, and it's something that we'd like to do, but we haven't done yet. But what you can notice is that the 20 carbon species, right, it's virtually absent in the cortex, but then it starts to upregulate very strongly in the stroke-induced area, at least on the periphery. So that was interesting. What does it mean? I don't know, but this is what we detected. The other thing that was interesting is that you started to see bilateral changes. You started to see bilateral increases of this GM2 or this GM1 20 carbon species in the hippocampus compared to the uninjured animal. Again, unilateral injury, bilateral response. And if you remember the stroke that we showed, where that unilateral subcortical stroke with the PET MRI and the bilateral inflammation, again, this is a very intriguing phenomenon, why an injury on one side of the brain will cause a bilateral response like this. Just more evidence that it will happen. But what's really interesting is that we were actually able to discover new targets. So these GM2 and GM3 molecules are precursors, and these actually, of interest, these molecules are associated with lysosomal disorders like Tay-Sachs and stuff like that. And so if you look at the uninjured animal, they're virtually absent, at least below our level of detection. And then between 3 and 7 days post-stroke, they're transiently expressed and then resolved. Again, at the border of the stroke-induced damage. So this was interesting to us. So this GM2, GM3 story is something that we're actually following up on more on a neuronal level, but without this MALDI imaging, we would have never been able to put the positional information in. And the fact that these GM2 and GM3 go up transiently, big deal, who cares? It's where they're going up that was really interesting. They're going up right at the border of the infarct. So right at that area of tissue that's vulnerable to cell death, right? Right on that border. So that to us was very interesting. So we're following some of this work up back in our cortical neurons, and it turns out that if you give GM3 to cortical neurons without doing anything else to them, that you can actually turn on. If you give glutamate, which is an excitotoxic response, so this will kill neurons for sure at 100 micromole. You give 100 micromoles of glutamate to neurons, you start to turn on GM3 expression. This is done through immuno. Again, recognizing that the limitations of the antibodies with these things, they are crap, but it does go from nothing to on, so that is pretty convincing for us. And then it stays on during the course of neurodegeneration. And the other interesting thing is, and I apologize, the light's coming, you can't see at the bottom, that if you just give GM3 to neurons and then look at apoptosis, so here you're looking at tunnel on the bottom, here you're just looking at general neurodegeneration with that PI stain. Again, you give GM3 to neurons, it kills them. So, so the question was, if you go back to here, one of the things that we weren't sure of, and the reviewers of my grants kept asking me, well, how do you know what kind of cell type these are expressed in? Maybe these are inflammatory cells, maybe they're neurons. Well, it does, at least based on this initial evidence, and this is data that just came out of the lab about a couple months ago, that it is happening at least in neurons. So we're encouraged about this. Why is this interesting to us? Because potentially these are novel targets for therapeutic intervention, right? And not only are they potentially novel targets for therapeutic intervention, because for sure GM1 has been tested clinically for stroke patients and failed. But why did it fail? Okay, ask the question, why potentially did it fail? When you give GM1 to a patient, you're just giving them a bunch of 18 and 20. You don't know how much 18 is in there, you don't know how much 20 is in there. Maybe these two things have actually completely radical different biological effects in a patient. So one of the things, actually we're just writing a grant and submitting it now to the Alzheimer's Society, is we are synthesizing just the 18 carbon species and the 20 carbon species of the GM1 and looking at its potential neuroprotective value in our Alzheimer's disease models. This GM1 story has also gained some traction. It's been shown to be potentially protective in Huntington's disease as well as in Parkinson's disease. So again, not just an Alzheimer's phenomenon, but across multiple neurodegenerative disorders. Okay, so again, here's some evidence that GM1 is important, and this is through a collaboration. So you give MPTP to induce Parkinson's disease in a mouse, and then here you're looking at the substantia nigra, and you're measuring GM1 levels. You have some GM1 levels in the substantia nigra. You give MPTP, you completely wipe out the GM1 levels. By 7 days, there's virtually none. At 14 days, and then we actually did some animals where we injected GM1 peripherally and found that we could get GM1 back into the substantia nigra, and then it protected these cells from undergoing cell death. So again, this is a useful technique, this mass spec imaging, not only for detecting native changes in membrane biochemistry or proteins or lipids or whatever, but potentially stuff that you actually inject into an animal and tracing whether or not it crosses the blood-brain barrier and all these different types of questions. And then what we're really excited about is being able to use this technology to image beta amyloid peptides. So one of the things that I started off by saying is that the amyloid field is extremely controversial. Okay, some people don't know if it's the 40 to 42, if it's monomeric, oligomeric, plaque. That's the problem. And so one of the things that we think we can do to help answer this is to use some sophisticated analytical chemistry to be able to look at what kind of beta amyloid. Is it oligomeric in form? Is it monomeric? Is it in plaque form? Is it 40? Is it a fragment? Is it the 3 to 17 fragment? Is it the 3 to 40 fragment? And be able to use the MALDI-TOF to do this. We have yet to be able to image it in tissue. So this is one of these work in progress. Imaging these things, amyloid proteins are extremely sticky, okay? And the way that MALDI works is you have to fire a laser, ionize a tissue, and then get the ions to fly it back. But because the amyloid is so sticky, it won't fly. Okay, so we are working on ways to make it fly. We can actually use this technique to pick up the A beta. So this is an animal, this is actually just beta amyloid that we synthetically made, and there are different protocols where you can take beta amyloid peptides and make them oligomerize. And we've used MALDI, and each one of these peaks is a beta amyloid species. So 4,000 is the mass units. So this would be like A beta 1 to 42. This would be the dimer, the trimer, the tetramer, and so on and so forth. And we can pick it all the way up to the 12-mer, so which is potentially one of the more toxic forms of beta amyloid. So we can do this stuff now in sample, and we're just submitting this for publication now. The next step that we want to be able to do is to use this MALDI to be able to image it. And why that's really interesting is now we'll be able to combine the power of some of the membrane biochemistry questions with some of the pathological questions. So now you'll be able to key in and use the positional information of where amyloid is being deposited and start to ask the question, what's happening at the local membrane biochemistry level in that area where you're starting to get beta amyloid deposition. So we're really excited about where this is going. We have a lot of work to be able to get it there, but that's kind of where we're at. I'm just going to skip ahead. So just to sort of summarize some of the stuff that I've talked about, and I realize I've sort of covered a lot of information here, but I hope you can appreciate that the work that we do in the lab, although there are two distinct themes, there's a lot of crossover between the two. So the first theme is primarily funded by CIHR, and the second theme is primarily funded by CFI and NSERC, but there's really a lot of sort of communication between the two, because a lot of the animal models that we're developing of comorbidity will be able to use for imaging for MALDI. And a lot of the targets that we're picking up in the MALDI we'll be able to use potentially as targets to intervene in our animal models. So thank you very much for your attention. The last thing I just wanted to say is that this is what happened like literally 3 seconds before the picture I took before. And so again, just highlights that a picture can tell 1,000 words. And speaking of pictures, I just want to sort of acknowledge the work done in my lab, because sadly I'm actually not in the lab as much as I'd like to be anymore. I spend too much of my time writing grants, which is sad for me. But I have a very talented postdoc who's come from the University of Alberta, and she's working hard in developing this transgenic animal model of Alzheimer's disease, and she's done a ton of a year's worth of behavior on an animal that hasn't shown any pathology, right? So talk about a huge leap of faith for her to come and do this work. But she showed some really interesting behavioral data that we'll hope to get out in publication in a couple of months. A talented PhD MD student who is doing some of the work on hypertension. Aaron's doing some of the work on stroke. Sarah's been a rock in terms of getting the MALDI going. So when I first came to Western, we didn't even have a MALDI. We had to go through collaboration on a MALDI that was 20 years old. And then we were able to get a CFI award to get a new one. So she was really instrumental in getting off the ground. Jasmine's a super talented student who's been doing a lot of the work on the beta amyloid and trying to bust her butt to figure out if there's a way we can image beta amyloid in the tissue. And that's not only just important for the animal work, but we also have clinical collaborators that have tissue from brain banks of Alzheimer's disease patients that we're just waiting to do some work on. And then Ryan and Dan, Jen, and Nadia are relatively new master students that have various projects ongoing in the lab. I just also want to acknowledge funding and also some of the teams. I'm on a CIHR and CCNA team on vascular cognitive impairment, as well as some of my collaborators in the mass spec world, and then Jay Schneider at Thomas Jefferson University, who is interested now in the ganglioside stuff on some of the Parkinson's work, and then of course my sources of funding. Thank you. I feel like Phil Donahue.
I just had a couple questions about actually the cyclodextrin
Yeah.
that was in the middle of the talk. So can you wash it out and get some reversal of the phenotype?
So we've tried that, and if you titer it down to a very, very small amount, the problem is doing the live cell imaging on it, right? Because the live cell imaging that I showed you over there was happening over 12 hours. So if you titer it down to like 1/1000th of the concentration that we're doing, it will sort of, we can sort of get the neurites to retract, but not fully retract. But we haven't found a way to get them to completely grow out. Like, so it's I think you can actually get them to regrow out, but we haven't been able to find that point of no return where you get them back to like nothing and then get them to completely grow out. So we're playing with it. We've also done this work, and I didn't show the data, but it was published because you could be asking the question, well, is it just a cholesterol phenomenon? So we used other agents like PDMP, which also impairs glycosphingolipid activity and lipid raft activity, and showed the same thing.
So this mass spec approach seems like a fantastic new tool to use, and I wonder, I mean, could you do things like make correlations between different diets of the rats and the ultimate outcome in terms of their susceptibility, and then make, you know, perhaps recommendations that humans could benefit from?
That's a great question, and that's exactly where we're heading. So one of my new master students, Nadia, is taking our transgenic rats and is giving them a high-fat, high-glucose diet, right? And then I just can't wait to get the brains of these things and do this imaging on them. I mean, that's the question. And the other thing is, I'm only showing you like sphingolipids and gangliosides, right? Once we image that database, we can go back and look at phosphocholine, phosphoserines. You could do anything. The issue with the MALDI is the sensitivity getting into the lower stuff. So when I did my postdoc, I published some work looking at like smaller glycerylphosphocholine members, so like platelet activating factors, stuff like that, which are kind of in the 300 to 500 mass units. That's not really good for MALDI. You need a more sophisticated, you can do it, and some of the new equipment, some of the new mass specs have the ability to image in those lower mass range. I can't do it in my lab, so it would have to be through a collaboration. But why that would be interesting is some of those smaller bioactive lipids might be, at the end of the day, the most important ones when it comes to. So it's been a strategy, because like from when I started at Western, I started with nothing, right? And so my goal was to get people interested in it, to get the CFI, to get the MALDI, to publish some work with the MALDI, and then get more people interested in it, apply for a second CFI to get more people in an institutional CFI so that we could go after some of the more expensive mass specs to answer some of the more sophisticated questions. But I feel like it's been kind of a ramping up process.
In one of your original slides, the beginning, you mentioned oxidative stress.
Yeah.
Have you done any work on oxidative stress and the correlation?
So one of our students who I co-supervise, Jen Au, works with a fellow out of Wayne State who's developed a catalase. He's taken the catalase molecule and tagged it to an SKL. So it basically targets catalase to the peroxisomes. And we've used, and it's a more efficient catalase molecule to reduce reactive oxygen species. Because what happens is, as you age, your peroxisomes fail, and then your catalase gets miscompartmentalized and doesn't go to the peroxisome, and you don't get the same effect. So we've used his agent in our animal models and shown it to be actually very, very protective. So her thesis work is looking at that, and she's following up by looking at like oxidative stress to lipid peroxidation, all that, those kinds of end products. Yeah, I think it absolutely does. And one of the things that, you know, this inflammation, going back to this inflammation story, is very interesting. Just got back from neuroscience, and this idea of microglia being in different stages of activation, whether even M1 versus M2, this ability to be in the pro-inflammatory versus the resolving anti-inflammatory state. Turns out that there's been a new receptor that's been identified as a major risk factor for Alzheimer's disease, and it's called TREM2. When TREM2 is mutated or knocked down in microglia, they can no longer clear beta amyloid. So what happens is that these microglia are still there, they're still spewing out reactive oxygen species and inflammatory cytokines, but they're not actually able to remove the material. And so it causes this exacerbated level of damage. So reactive oxygen species are just as important, I think, as the inflammatory response. We just looked at the inflammatory response. All right, thank you.